We're actually going to start this lecture on the chapter as opposed to my outline. The only real section of the chapter that we're thinking about right now is chapter 4, section 15, endosymbiotic origin of mitochondrial chloroplasts. There's a nice little graphic that shows some type of nucleated cell with endoplasmic reticula engulfing an oxygen-using prokaryote, this thing over here. I don't know if you can see my cursor, and the mitochondrion becoming that, okay? And then later on, at that point, you've got a non-photosynthetic eukaryote, and at that point, we're going to be engulfing a photosynthetic prokaryote, it's kind of over here, which is eventually going to become the chloroplast. Now, I think this diagram is completely Gumby and almost to the point of being a falsification of our best known model of how chloroplasts and mitochondria evolved. For example, there's no evidence that the cell was actually nucleated at the time this happened. It probably wasn't, right? And so while there's a nice brief and unsatisfactory discussion of endosymbiotic origins of mitochondria and chloroplasts in the textbook, I'm going to go ahead and work with my own version, which is something that I think I can do better than what the textbook is doing, right? Now, the bottom line here, it's actually pretty, you can kind of go at the end result. The objective here is for you to really get that mitochondria and chloroplasts, different from other organelles. I mean, thinking about all the organelles we learned about in the previous lecture, the rough ER, the Golgi, the nuclear envelope, the ribosomes, they're all different from mitochondria and chloroplasts. It's like these two organelles are special because of the way they originated, right? And it's a pretty simple story even there. Okay, one, mitochondria started out as free-living bacteria living on their own as bacteria, and then they got taken inside of a bigger cell, a different cell engulfed them, right? And over a long period of time, they eventually became mitochondria, right? Chloroplasts, our second organelle with a similar origin, were once free-living photosynthetic bacteria. We call them cyanobacteria. And cyano means kind of a blue-green color. So these were pigmented bacteria. They're able to use light energy to convert carbon dioxide into sugar. And that was an important biochemical trick. The cells that were able to engulf those cyanobacteria then all of a sudden took advantage of the ability to make food out of the air that we breathe, okay? And so chloroplasts are also really, bottom line, they're actually photosynthetic bacteria, only they've been living inside of a larger host cell for a really long time. They've evolved, they become completely dependent on the host cell has evolved to become completely dependent on the chloroplasts. The same goes with mitochondria. And we say that this type of origin is given the name endosymbiosis, which literally means, well, symbiosis means living together, right? Endo means within. And so literally within, living together, and in particular, one cell is living inside of a different cell that we call the different cell the host cell. Now the host cell tends to be larger, to be the host, and the endosymbiont, the cell that's living on the inside, is going to be living on the inside. Maybe this is the one that's going to become the mitochondria, right? So this would be the endosymbiont. And this will be the host, right? And over a long period of time, the endosymbiont becomes the mitochondria. Whereas the host becomes the actual eukaryotic cell. And so that's the basic concept of endosymbiosis. In the case of a chloroplast, we have a cyanobacterium that goes inside of a host cell. It's the cyanobacterium endosymbiont. And that's one that will eventually become the chloroplast. Okay? So in the big picture, you want to understand what endosymbiosis means and the idea that it only applies to these two organelles, mitochondria and chloroplasts. Okay? The next thing that some of you who might be skeptical would want to hear about, how do we know this is true? Is this something that has evidence, or is it simply a story? No, it has a huge amount of evidence. I mean, the evidence is so strong, you really, can't argue with it. I mean, what would you expect to find if a chloroplast was actually derived from the cyanobacterium? I mean, wouldn't it help to find a genome inside? Okay, so if there's a DNA genome, it's a circle in a circular chromosome. And when you look at the genes inside of the genome of the chloroplast, they actually match up really closely with what you find in free-living cyanobacteria today. In other words, we're actually seeing, as a result of a genetic analysis, a familial relationship between cyanobacteria and chloroplasts that live inside of plant cells. Okay? But same thing goes with mitochondria. There's a genome inside of that. The genome, remember, the genome is kind of like the sum total of all the genetic information that's there inside of the structure. I'm not talking about the genome that's inside of your nucleus. There is a nuclear genome as well. The host cell has its own nucleus, and the nucleus has its genes. Separate from the genes that are inside of the nucleus, there are actually genes that are inside of the mitochondria, and they're in a circular chromosome. And the genes that are there inside of the circular chromosome of the mitochondria match up pretty closely with what you find in E. coli. Okay? So yeah, I mean, if you were to find a free-living mitochondrion out in the world today without the rest of the eukaryotic cell, through a simple DNA analysis, you would place it within the bacteria, right? within that group of bacteria that is most closely related to the mitochondria, right? If you were to find a chloroplast without the rest of the cell and looking only at the DNA inside the chloroplast, you would classify it as a cyanobacterium, right? So yeah, cyanobacterium, genetic similarity with the prokaryotes from which those organelles are derived, okay? So that would be a pretty powerful set of evidence on its own. Second pretty compelling set of evidence is that these organelles, they have their own ribosomes, right? The ribosomes that you find inside of a chloroplast are similar to the ribosomes that you find inside of the mitochondria, which are similar to the ribosomes you find in either cyanobacteria or in other free-living bacteria today. In other words, you have the 70S ribosomes that are characteristic of all prokaryotic ribosomes. And those are the same ribosomes you find inside of inside of a chloroplast and inside of a mitochondria, right? The ribosomes you find out here are actually much bigger. The ribosomes in the eukaryotic cell are bigger, 80S. And qualitatively different from the bacterial ribosome, and the bacterial ribosome is actually really similar to the ribosome you find inside of mitochondria, and hence, yeah, looking at that, oh, you know what? Mitochondria have bacterial ribosomes. They've got a circular chromosome. The genes inside of the mitochondria look like the genes you find in E. coli. That is, that's all evidence that stacks up in favor of the idea that mitochondria take their origin from free-living bacterial cells. Okay? Okay. And one little piece of evidence beyond that is that if you look at the size of bacteria, maybe this is a bacterium, and this would be a mitochondrion, the size is really similar. And there's structural similarities. Mitochondria have two membranes, bacteria have two membranes, right? And so structurally they're similar. When you look at the way that they reproduce inside of a host cell, they reproduce on their own independently of when the cell itself is dividing, right? So we'll talk about how cell division is the coordinated thing, how all the parts of the cell are dividing at the same time. The exceptions to that are mitochondria and chloroplasts, which divide on their own timetable, right? In other words, they've got kind of like a reproductive independence from the host cell on its own, okay? And so I think that the evidence supporting the idea that mitochondria and chloroplasts really are not the same as what goes on inside of the regular cell, that's a pretty satisfying story. Okay? Now, the thing that I want to develop with you, and this is going to be separate from what you have in the textbook, is the narrative, the story, how it is that mitochondria took their origin. And this requires us to go back in history, our Earth's history lesson. Which is going to take us on to a different slide. And this is going to be giving you a little bit of a understanding of the bigger picture of biology. This is kind of like what I talk about at length in my other class, BIO110. And so if this is going to be a timeline, right? Right here would be the present, right? And then if you go back in the present, it should be a line. That's going to be 1 billion years. 1 billion years ago. 2 billion years ago. 3 billion years ago. 4 billion years ago. And it goes back, the Earth goes back as far as roughly about 4.6 billion years. That's how old the Earth is. The solar system is about the same age as the Earth, and it goes back 4.6 billion years. The actual universe is about 12.7 billion years. And so you can take a cosmology course and learn more about that. Okay? The reason why I'm putting the geologic timeline up here is because we've got a lot of evidence supporting the idea that right here around 2.5 billion years ago, we have the great oxygen revolution. Okay. In other words, before this time, there's no oxygen, no O2. Remember, oxygen is that molecule. When I say oxygen, it refers to the molecular oxygen, not the atomic oxygen. Certainly we had oxygen, the element, before that. What we're lacking before that is oxygen O2. Okay. After that, we have O2 present. Okay, so from 2.5 billion years to modern times, oxygen, this gas that we have today at around 21%, was there. Not necessarily the same abundances. It's fluctuated. Initially, it was very, very low, but the fact that it was present is the key feature to the story, right? And the reason why we care about this is that the host cell is presumed to be anaerobic. Okay. The cell that eventually became the eukaryotic cell was anaerobic. And as it's moving across this boundary, it gets itself into the early days after the oxygen revolution. And it is really, really sad because oxygen, O2 has the ability to oxidize proteins and destroy the proteins that are there inside the cell. The host cell was actually suffering by the presence of oxygen. And the only way that the host cell was able to persist was by taking in a bacterium that was aerobic. And the aerobic bacterium evolved and was able to take in oxygen gas. And one of the things we sort of visualize here is that as a result of taking in the aerobic bacterium, the host cell was all of a sudden able to deal with the presence of oxygen because the aerobic bacterium was absorbing the oxygen that was otherwise causing damage, right? This here was not allowed to happen because the aerobic bacterium was consuming the oxygen before it had a chance to destroy the cells, membranes, and proteins, and DNA, right? Now, the reason why this is important is because in the history of life, prokaryotic cells start off around here, around 3.5 billion years ago, the origin of prokaryotic cells. Yeah. Which is also the origin of life, right? Because life includes the prokaryotic cells. The origin of eukaryotic cells. Origin of eukaryotic cells does not take place until after the oxygen revolution. And one of the features, maybe even more important than a nucleus for eukaryotic cells, is the fact that they've got this endosymbiotic bacterium, which is serving the role of being a mitochondrion. We associate the origin of eukaryotes with the changes that occur right here at the Great Oxygen Revolution. And that's your little piece of history lesson for today. Your cells start right here. All of life starts right here. One thing that happened to your cells, to the history of your cells, was that they took their origin by combining a prokaryotic cell with a larger host cell to give rise to this process of endosymbiosis. Which accounts for the origin of mitochondria. Okay. Chloroplasts come in at a later juncture. Not all cells have chloroplasts. This is our eukaryotic cell. Eukaryotic cell. Here's the mitochondria. Remember, we don't have a single mitochondria. There are dozens, sometimes hundreds of mitochondria. Okay. Which are actually bacterial cells. Bacterial endosymbionts. The idea here is that by taking a chloroplast, or taking a cyanobacterium, inside, letting it turn into many of these green cells. Some chloroplasts are here. At this point, the cell is ecstatic because not only does it have the ability to deal with oxygen, doesn't have to be killed by the oxygen. The chloroplasts are taking in the light energy and releasing food into the cell. So effectively, the cell doesn't even need to eat anymore. right? We've taken care of all of our basic needs by having these endosymbiotic bacteria and cyanobacteria living inside of us. And that's what makes plant cells so successful. It's kind of a neat story. Now let's go back to our original share. Okay, and just clarify that we've covered this item here. Okay, the history lesson allows us to think about that endosymbiotic origin of eukaryotic cells as associated with the great oxygenation, the great oxygen revolution, right? And I've mentioned this before, but the other organelles like the endoplasmic reticulum, they're not associated with endosymbiosis. And again, there's a reason why I'm pointing this out, and there's a source of confusion. You've got the endomembrane system, which ironically are not associated with endosymbiosis. So if you are in the habit of basically associating words by similarities, don't do that here because it's going to lead you to the wrong answer, right? Endomembrane system are not originating from bacterial cells that live inside of a host cell. Bacterial cell living inside of a host cell, that is endosymbiosis. And you want to make sure you have that idea clear and distinct from the concept of an endomembrane system. Cell inside host, right? Not the same thing. Got it? Okay, next time we start talking about, I'll have to see what we have to talk about, but we're getting close to metabolic biochemistry really soon.